Hello everyone. So my name's Sam and I'm a neuroscientist working at the Florey Institute at UniMelb. Quick disclaimer, I have no cool props. I apologise, I'm not going to be lighting any light globes alight. But hopefully I will be able to dazzle you with the wonders that is stem cell biology. So firstly, let me introduce you to what I mean by diseases of the brain. So this can mean neurological deficits or diseases like Parkinson's and Alzheimer's disease. It can mean mental illnesses like depression or schizophrenia, and it can also mean substance abuse like drug and alcohol addiction. And actually 1 in 6 people will be affected by one of these brain diseases or disorders in their lifetime. So it's quite a significant burden on our society. If we take substance abuse out of the equation, brain diseases and disorders cost over $31 billion annually in Australia. And actually over the next couple of decades, they're on track to overtake the cost of heart disease, cancer, and respiratory diseases combined. Now this is due to a few different reasons, but one of the main ones is due to ageing. So Australia and most of the developed nations are ageing populations, so people are living longer and there's more older people. And so this means that there's increased prevalence of diseases like the dementias, Alzheimer's, Parkinson's, Huntington's. And so it may surprise you to hear that actually we don't know a lot about these diseases. We don't really know what causes them, we don't really have a lot of treatments for them, which is pretty terrifying. But we need to be realistic. If someone has cancer, you can biopsy the cancer, you can cut it out, you can study it. If someone has kidney disease or liver disease or lung disease, we can biopsy those organs, we can even give them whole new organs. Whereas the brain, we can't really cut into, it's very difficult. And we certainly can't give people brain transplants, despite some people needing them. And so what this means is that we're quite limited in our capacity to actually look at the brain and see what's going on. But some of the things that we have got my pointer, so we've got MRI images and we've got DTI images here. And this is our best way of looking at what's going on in the brain. But if you think of someone that comes into the clinic and they've got symptoms of dementia or Alzheimer's disease, they'll get one of these scans, and these scans are great. Doctors can read these, they know exactly what it means, they can usually give you a general prognosis. But with a disease like Alzheimer's, what we know is actually some of these the disease can actually begin up to weeks, months, years, even decades before that symptom onset. So that's actually quite a large window between when the disease actually begins and when the symptoms come on, and we don't really know what's happening in that large window. So I'm going to make you all neuroscientists in this one slide. This is my 3 years of science undergrad in one slide. This is you. Here is your brain, your spinal cord, and your nerves. And these nerves go off and innervate your organs and your muscles. If we add another layer of complexity, this clinically relevant blue blob is now your brain. And we know that the brain has 10 billion nerves inside it, and each nerve is a messenger cell that passes the messages around your body. So here's an example of one nerve. Here's the cell body, the axon going down into your spinal cord. It forms what's called a synapse with this cell body. The axon then goes out of your spinal cord and it goes to your muscle. And so your brain has gone down your body to say, let's wave. Let's add another level of complexity. Your nerves can come in all shapes and sizes. This example is a motor nerve, so one that innervates one of your muscles. And what you can see, we've got our cell body and our axon, like in our blue blob here. What you can see is all of the dendrites around the top here and at the bottom here. And each one of these dendrites can form the synapse with a different neuron. And each one of those can form synapses with other neurons. So you can see that this is rapidly becoming a very complex system. But it actually gets even more complicated because nerves aren't alone in the brain or the spinal cord. So now we have our nerve here in the yellow going along here, forming our synapse with the next neuron. But actually the neuron is completely ensheathed with all these different cells that are trying to help it stay happy and healthy and functioning. So we've got our blood vessels surrounding it, we've got our astrocytes that form the kind of structure that glue everything in place. We've got our oligodendrocytes that form this insulating sheath down the axon. And then we have our microglia, which are kind of the garbage trucks that eat through and survey the brain and keep it happy and healthy. And so in the context of this, you can see that our MRI images and our DTI images don't give us nearly the amount of resolution to work out exactly what's going on. And so in our neurological diseases, it could be one or all of these cells that are involved in this process during that window when the disease starts to when the symptoms come. So I'm being a bit pessimistic, and I think I'm trying to make it sound more exciting so you think the stem cells are amazing. We obviously have other models to look at these diseases. So we've got things like post-mortem tissue, and we've got a huge number of animal models to interrogate these systems. But the drawbacks are that in post-mortem tissue, again, we're looking at end-stage disease. These people have died from the disease, so we're missing that kind of integral window. Animal models do let us look into that window prior to the symptom onset, but mice don't get dementia, so we're artificially causing these symptoms in this animal to try and interrogate what's going wrong. So we ideally need a humanized system. And so this is where the stem cells come into it. So what is a stem cell? Mr. Sperm and Miss Egg become fertilized and we have our embryo. And so within the embryo are these blue cells that are called stem cells. And what this means is that they have the capacity to become any of the cell types of the body. So we have our master stem cell, and then they can pick which lineage they want to go down and what type of cell they want to become. And what we know is that once the stem cell, oops, daisies, I gave away the answer. Once these stem cells decide what they want to become, they can't actually go back to becoming stem cells. So this doesn't really help us interrogate what's going on in our brain cells, because we can't isolate any of these cells or make any of our neurons. And there is still the kind of ethical considerations tied with embryo stem cell research. So then along came this guy, Shinya Yamanaka. He was a Japanese scientist who won the Nobel Prize in 2012 for his brilliant idea. So what he did was he took a patient with whatever disease we want to look at. He cut out a small piece of their skin, cultured their skin cells, and then added this cocktail of factors that perfectly turned on and off the right genes that actually re-derived these skin cells back to these stem cells. And so these are known as induced pluripotent stem cells. So induced, we made them. Pluripotent, they have many potentials, and then stem cells, they can become any cell type. And so magically, this guy has gone from having an adult person where, remember, none of these cells can go backwards to becoming stem cells, and he's magically made them go back to stem cells. And so this finally gives us a way to actually look at brain cells and interrogate what's going on in the brain. So a wise man who's in the back of the room once told me things are best explained in threes. So I'm going to be telling you how induced pluripotent stem cells can be used to address the gaps that remain in neuroscience. The first one I'll talk about is how we can use stem cells to understand the causes of disease. The second one, how we can find a treatment for disease. And the third one, how we can use these stem cells as the treatment for disease. And so for each one, I'll give you a specific example, but trying to kind of extrapolate it to other diseases and try and see the true potential of these stem cells. Okay, so the first point, understanding the cause of the disease. To explain this, I'm going to use the example of motor neuron disease. So MND is also known as amyotrophic lateral sclerosis, ALS, or Lou Gehrig's disease. It's characterized by the progressive death of the nerves that control your muscle movement, and these are called motor neurons, which I introduced earlier. And they're located in the motor cortex, which is around here, and all down your spinal cord. And they're the nerves that are important for innervating your muscles. So we get death of those nerves, you get denervation of that muscle, and then you get the muscle atrophy. And that's why patients with MND get lost the ability to move, to walk, and they even lose the ability to breathe, swallow, speak. So it's an awful disease. It's not overly prevalent, but it does have a very short life expectancy. So it truly is a rapidly progressive, debilitating disease. But what we now have the capacity to do is use what Yamanaka did and take our patient that has MND, take their skin cells and make induced pluripotent stem cells. We can then take our healthy patient and do the same thing. And what this means is effectively we have these two patient cells in the dish, and we can determine how they differ, why they differ, and how we can make the MND patient's cells more similar to the healthy patient. So this is what the cells look like in the dish. Each one of these little dots is a cell, and they grow really happily in these little clusters called colonies. So then we turn on and off certain genes, and we start directing these cells towards becoming brain cells. So these are showing positive markers for genes that are responsible for your brain cells during development. And then what we can do is we can start addressing the complexity of our system, and we can start making the different types of cells. So we can make our motor neurons, our astrocytes, our oligodendrocytes, and our microglia. And what's really cool about this system is that we can look at the motor neurons from our MND patient compared to control and work out why don't they live as long, why aren't they as happy as healthy. But what we can also do is add in, say, astrocytes from a control patient to the motor neurons from an M&D patient and say, do these astrocytes make the motor neurons happier? And if yes, why? And is that a factor that we can target with a drug that potentially could actually benefit a patient? So it really is a really powerful system to tease out exactly what's happening. And then the next level is that we can then start combining these cells to form mini brains. And actually that gives us the capacity to kind of form a more complex structure and tease out again what's going on and try and add drugs into this system to fix the deficit. So I've got one slide to tell you specifically about my research, so I'm going to be really excited. I work on this cell type called the oligodendrocyte, which is the coolest cell in the body. So we've got our motor neuron that I told you about. The role of the oligodendrocyte is it forms this fatty sheath around the axon. And so much like a power cord that has the rubber around it, it's important to kind of maintain conduction velocity and keep the nerves firing quickly and without crossing of the wires. What it also does is the oligodendrocyte can feed the neuron through this myelin sheath. So if you think about, say, your nerve going from your brain to your spinal cord, or even your sciatic nerve from your spinal cord down to your foot, these can be huge neurons, and so they kind of heavily rely on this sheath to keep them happy and healthy. So, most important cell in the body. So what we do is we take our stem cell from our M&D patient and from our control patient. We make our oligodendrocytes, and then what we can do is we can study these and work out how they differ. So one of the assays I did was look at their capacity to produce fuel to traffic into and feed the neurons. And so we've got oligodendrocytes from 2 M&D patients, and you can see that they're quite low. They're about medium in their capacity to produce fuel. When we can compare it to the controls, you can see the controls have a significantly increased capacity to produce fuel. So what this means is that oligodendrocytes in patients with M&D have a reduced capacity to feed and keep the neurons happy and healthy, meaning that they are contributing to the disease. And what this means is as we tease out these pathways in the different cell types, we're providing therapeutic targets that we can then find a drug and try and target it to try and increase this capacity up to the control levels. And so this then ties into my next point, is how we can use stem cells to find a treatment for disease. And again, I'll use M&D as my example. So again, we're going to take our stem cells from our M&D patient. And I made a little video. This is what, this time we're going to make motor neurons. And this is what they look like, maybe, growing in the dish. That wasn't really worth it, was it? But they're really cool. So they grow over time, and then we have our motor neurons at the end. And so what we can do this time is that we can plate these motor neurons into each of the wells of our 96 well plates or our 384 well plates. And it means that each well will have an identical amount of cells in them. And then we can add a huge number of drugs, where we can add 1 to each well and work out, does drug A make the motor neurons happy? Does drug B make them happy? And try and work out how and why they're working. And so maybe this one will be more impressive. And so what's really cool is there's a part of our lab where we have this all set up and it's highly automated. So this means that we can screen thousands of drugs within a couple of hours. So we truly have the capacity to look in high throughput at what drugs could be working. So our robot has taken the plate out of the incubator and put it here, and you can see that we're able to feed the wells and put drugs in, and it's all completely automated. We can program it into the computer. And we're very lucky to have this funded by the Vitamin D campaign, and so it's a really exciting time and shows great promise for the field. Oh, watch it again? No. Okay, so finally I'm going to touch on how stem cells can be used as a treatment. And so the first 2 points are actually the research we do at the Florey, but I've delved outside of the field to cover this one. So for this third point, I'm going to talk about Parkinson's disease. So Parkinson's disease is characterized by the progressive death of nerves that are responsible for relaying the messages that plan and control your body movement. These neurons are called dopaminergic neurons, and they're located in your basal ganglia, which is located around here in your brain. It's a really small area. The 4 key symptoms of Parkinson's disease are tremor, slowness of movement, muscle rigidity, and increased likeliness to fall. And Parkinson's disease is much more slowly progressive than motor neuron disease, but it still is ultimately fatal. And it's actually much more prevalent in Australia and actually worldwide than motor neuron disease. So to cover this point, I'm going to take you back to the 70s, 80s, and 90s to show you what the original research was. So scientists took what were aborted fetuses, isolated the brains, they found the basal ganglia, they isolated the dopaminergic neurons, and they transplanted these into patients that had Parkinson's disease. And then they used immunosuppression to stop these patients from rejecting the cells. So this is obviously extremely controversial, and it did cause a lot of issues at the time and even now. But what you can see is that the concept actually is really effective. So in the white line here, this is Parkinson's patients that did not receive a transplant. So this is over a 2-year window, and you can see that over time they had the slow progression that's characteristic of Parkinson's disease. In this next study group, you can see they had this improvement over the first year, but then returned back to baseline, whereas these two showed a significant improvement in motor function. I don't know why negative is an improvement, but if it goes down, that's a good thing. And so it shows that this truly does have a lot of promise as a concept, that if we can replace these cells with a healthy alternative, theoretically it has the capacity to improve the symptoms of these patients. So now if I introduce the induced pluripotent stem cell, we have a more ethically friendly alternative. So we have our patient that has Parkinson's disease, and we can actually isolate the skin cells from the patient themselves, which means that we won't need the immunosuppression because they won't see it as a separate transplant. We then make our stem cells and change them into these dopaminergic neurons, and we can actually tinker with these neurons if we need to kind of optimize them so that when they're transplanted, they're truly effective in rescuing the phenotype. And actually this has happened for the first time last year in Japan, so stay tuned, I'm sure the results of that will be coming shortly. So I've got 2 slides left, and I wanted to talk about kind of the pros and cons of stem cell research. So I'm sure a few things that I've said along the way may have kind of triggered a few thoughts in your mind, because it is a really quite a risky field to be working in. There is a lot of potential for it to be abused. So I want to finish on the positive, so I'm going to start with the negatives. I'm going to talk about a couple of potential drawbacks of stem cell research. So the first one is the risk of eugenics. I don't know how many of you have seen this classic Gattaca, but it perfectly illustrates the risks of tampering with human DNA and kind of the risks of things like, why have I completely forgotten the word, where they make the babies. What am I looking for? They alter the babies. Designer babies, thank you. Crowd interaction, there we go. The risks of designer babies, because currently with things like IVF, we can pick and choose the fatal genetic mutations and exclude those. But kind of where do we draw the line? Can people start choosing about gender or height or IQ? It really is sort of it's a murky line that hasn't really been drawn yet. The second risk with stem cell research is forced evolution. And this kind of comes back to the example I just told you. It doesn't seem like ScoMo is going to quickly throw this onto the PBS. It's going to be a really expensive technique, isolating someone's stem cells, culturing them, checking them, genetically altering them, and transplanting them. And so this means that it's only going to be available to a subset of people that can afford it. And this means that actually the survival is going to be based on wealth rather than anything else. And so it again is a potential drawback. And then finally, the third one, of course, is the risk of use without appropriate testing. And this one's pretty self-explanatory. Already, there are some countries around the world that have rapidly put stem cell transplants into the clinic without suitable testing. I'm sure many of you have read about the Chinese scientist who's genetically altered the embryos for fathers who have HIV, but had no ethical checks before he did that. So it certainly is a risk that, with this power comes a bit of a risk of people abusing it. But stem cells are awesome, so as long as we do carefully regulate this technique, it truly does have the capacity to work wonders in our field, particularly in neuroscience where the brain is something that's so difficult to model. And so I've already talked about these top ones here, the testing of drugs and studying cell-to-cell signaling and cell biology to tease out disease mechanisms. But separate from the brain, it has many other possibilities as well. We can make bone marrow for leukemia and chemotherapy patients. We can make heart muscle cells for heart disease, and we can make pancreatic islet cells for diabetes. And this is just a few examples of things, so it truly is a really exciting time to be a stem cell biologist. And with that, I'd like to thank you.
Thanks so much, Sam. We'll take a few questions. Yep.
I'm curious about what's the timeline from first with motor neuron disease and the introduction of stem cells before there's proof that this is a good idea. What was it, a couple of years?
What's the timeline for fixing diseases like MSD?
So actually using the cells as a transplant for the patients. It's a tricky one. MND in particular is a hard one to do trials because they have that rapid disease course. So actually it's both a pro and a con. It makes MND patients difficult for clinical trials because we don't have a large window to look. But at the same time, because it's such a debilitating disease, people are generally faster to sign up for things like stem cell transplants. So I would say probably in the next 5 years we're looking at the introduction of, I don't want to be hasty, maybe 5 to 10 years. It's certainly a rapidly advancing field, and you know, in Australia with Neil Danaher, and there are people like him all around the world raising awareness. MND certainly receiving a lot of attention and a lot of funding, which is great.
5 to 10 years. Over there. Yeah.
Hearing loss? Stem cell transplant and hearing loss. Oh, God.
Good question.
Very good question. So I'd be completely making it up. I do know that there is research going into the cochlear transplants, but kind of adapting them so they have more biological basis. But I don't know a lot about it, sorry.
It's a whole other Nerd Night talk. We'll take a question from up front.
If someone was to create a design baby or modify something,
Yep.
if you looked at that person later, would you be able to tell that there was modifications done, or would you hear that there was an actual reveal?
Can you tell if someone's a designer baby? You put it on your Tinder profile. Sorry, go ahead.
Look, it depends on the mutation. At the moment, I use designer, well, I didn't use, you had to tell me what the word was. Designer baby, I use it quite loosely because at the moment we can only screen for kind of congenital genetic mutations. So when I bring you back to the example of the Chinese scientist, so his idea was that there were fathers with HIV, and so in the embryos he genetically engineered them to be resistant to having HIV. And so it's a brilliant idea, but there was no ethical standard, so no one knows if he has switched that gene off, what are the downstream effects of that? And it's the way, you know, with everything, we've got billions of genes, so how do you know if you alter one that you're not going to subsequently affect anything? And that brings me back to Gattaca, the excellent movie, is that they did have designer babies to pick the best ones. It is a fictional movie, but still, the concept is correct. And it had downstream effects, and so they had other abnormalities associated with the attempted corrections.
We'll take a couple more questions. One from the back. Can we get some house lights, please, so we can see the people at the back too?
So can stem cells be used as a preventative rather than a treatment? For example, MND, if you know there's a hereditary, speak to the family who've got that certain gene, can you take stem cells before it occurs, or
Repeat the question, Sam.
Yes, so the question is, can we use stem cells as a preventative measure as well? And the example is MND. I don't know about MND. As an answer, I'll tell you about cystic fibrosis, which they know is caused by one gene mutation. And so they are trying to find a way to deplete the mutated cells and kind of bring in the new ones that don't have that genetic mutation to kind of replenish and take over to hopefully rescue that disease. So I guess the hope for MND is that potentially they can do the same, but there isn't as much of a genetic cause of MND as there is for diseases like cystic fibrosis.
Yep. Any other questions? One over here.
Yes. So you're asking about kind of the heterogeneity of disease?
Yeah, like MND,
Yeah. Yeah. Absolutely. Yeah, so a disease like MND has lots of different causes. 90% of it is sporadic. It's sort of a random onset. We don't know what causes it. With the movie I showed you where they're screening the drugs, actually the funding supports the kind of attempt at personalised medicine. So they've got stem cells from 150 MND patients, and they'll screen drugs on all of them to try and work out what works for what patient, and then try and regroup them and see if they can classify them into smaller subsets. So there are attempts to try and subclassify even in the random diseases.
Take one more question over here. Yep, you. Yep. Oh, there's two. All right, we'll do the person in the front, and we'll do the one at the back.
Really easier question.
Because you brought up the question about the drug screening. When you're doing the drug screening on the automated one, you just test the types of drugs or also the quantities of drugs that would be useful to treat the disease?
So you're asking about the drug screening, how we do it. So actually the FDA library in Australia has thousands of drugs, and so at the beginning because it's high throughput, can do all of them, and then you kind of pick out the best ones and then do a dose response for those and then kind of tailor it in, and then you can add in more patients, more cell types, and really try and work up to getting it into a clinical trial.
We'll take one last question, person at the back.
With the autologous transfer, where you're taking somebody, the Parkinson's example again, if you give somebody back stem cells that have still got the disease, don't you end up back at the start?
Yes.
This
Effectively.
Plus minus equals.
So I've got
minus?
two answers to that, because what I think is cool and completely unrelated to your question. In the graph I showed in the bottom left, actually even the ones that had the significant improvement ended up getting Parkinson's. So the disease ended up kind of infecting the transplanted cells. So we do need to tinker with it more. That wasn't your question. So the example I gave on the right that they did in Japan last year, they did use a third-party donor. Because you can still match people quite closely, so there is a possibility. The idea is eventually to be able to do autologous transplant with the patient, and we have the capacity to tinker with it and genetically modify it to hopefully eradicate the disease, but it is further on than what it is. I don't know if the Japan study, I think we're still a bit of time before that becomes kind of a worldwide phenomenon.
Great. Well, thank you so much for being